We're very happy to have Dr. Milton Pividori, who's coming to us from the University of Pennsylvania, or he's a postdoc with Marilyn Ritchie. He's previously done some work in the Green Lab, and before then was a postdoc with Hakim at the University of Chicago, and did his PhD in machine learning and computer science back at the National Technological University in Argentina. He's worked on a lot of exciting approaches involving omics and deriving latent factors from genomic data. And so we're very happy to have Milton speaking today. And there was a request to wait until the end for questions, so we'll circle back at the end for questions. But thanks again, Milton, and the floor is yours.
Thank you so much, Chris. That was a kind introduction. Thank you, everyone, for attending today. I will be talking about interpretable computational approaches to precision medicine. I'd like first to add a little bit to what Chris said about my research journey. I received training in informatics. During my PhD, I applied machine learning methods, in particular unsupervised models, to bioinformatics. Then I moved to the University of Chicago, where I worked on genetic studies of complex traits with Dr. Hakim, as Chris said. Then I moved to the University of Pennsylvania, where we are developing methods for omics data integration. And that's how I became a biomedical informatician. In this talk, I will briefly introduce genome and transcription-wide association studies, GWAS and TWAS. I'll show you how we apply these approaches to understand the mechanisms of asthma. Then I will walk you through Phenoplier, which is a method that integrates TWAS with gene co-expression networks. I'll show you how it can improve prioritization of candidate targets, the drug disease prediction, and the identification of shared mechanisms among different traits and diseases. Then something about my future research in developing portable polygenic risk scores. So precision medicine is an emerging approach for disease treatment and prevention that takes into account the individual variability in genes, environment, and lifestyle for a person. So these three factors are really important in understanding disease. In genetic studies, we want to find these genetic risk factors that lead to different diseases. Genetic studies can contribute in two broad areas of precision medicine. First is the identification of potential therapeutic targets, understanding of the biological mechanisms underlying disease. And the second area is the prediction of individuals at risk of disease. The most popular methods is genome-wide association studies, or GWAS. Here we have the genotype data for a set of individuals, and we have a trait of interest. And what we do is to associate each of these SNPs with this trait. And the output basically is a table where we have the effect size and the p-value of association between the SNP and the trait. The way we summarize the results in GWAS is using a Manhattan plot, where each point here is a SNP located in a particular region of the genome, and it shows the strength of the association. However, with GWAS, we only find the statistical association. We usually need to perform some extra analysis post-GWAS to understand the mechanisms mediating that GWAS association. One of those methods is transcription-wide association studies. Here, what we need is a data set like GTEx, for example, where we have genotype and expression data. So we build prediction models of gene expression using that genotype information. The nice thing here is that then we can apply TWAS in cohorts without expression data. So we predict the expression of our individuals, and then we correlate that predicted expression with a trait of interest, and we obtain these results that are now gene-based. So TIWAS tests whether changes in gene expression mediate the association of variants with traits. And this gives us a more mechanistic understanding of GWAS disorder. So let me show you how we apply these methods to understand a particular disease. In this case, it was asthma, and it was done during my stay at the University of Chicago. Asthma is really an umbrella term that comprises different but also overlapping syndromes. For example, the age of asthma onset is a key discriminating factor in identifying asthma phenotypes, and they differ in many aspects. For example, adult onset asthma is more common in women. Prognosis is usually worse. It has different responses to treatment, and it's less allergic. So the goal of this study was to understand whether this phenotypic heterogeneity reflected differences in genetic risk factors and molecular mechanisms. For that, we used the UK Biobank. We defined these groups, mainly childhood onset and adult onset asthma, with different sample sizes. These are the GWAS results for childhood onset asthma with blue at the top, and the results of adult onset asthma in red at the bottom. This approach allowed us to identify genomic loci that were specific to one of these phenotypes, like this one in chromosome 17, which was already known by the time of the study. We also identified a region that was specific to adult onset asthma, as well as different regions that were shared between the two. For example, here we found two independent loci in the HLA region. So this first study and results showed us that the genetic risk factors, if you take a look at this Miami plot, the genetic risk factors of adult onset asthma seem to be a subset of those of childhood onset asthma, with fewer significant loci, generally smaller effect sizes. So this first suggested a greater role of environmental factors in adult onset asthma. So that was the GWAS part of the study. We also wanted to test whether changes in gene expression were mediating these GWAS associations. For that, we used TIWAS. We focused on 5 tissues of interest. These are the results in skin. In this plot, each point is a gene colored according to the region of the genome. The x-axis has the significance of childhood onset asthma, and the y-axis the significance of adult onset asthma. And the different dashed lines represent different thresholds. So for example, PGAP3 has a positive Z-score. That means that a higher predicted expression of this gene is associated with asthma risk. However, this SDMA gene, which is also from the same region in chromosome 17, it has a negative Z-score, meaning that a lower expression of the gene is associated with higher risk. So these different patterns show how the SNPs in this region have a broad and complex regulatory effects. That was an interesting finding. We also found many genes that were shared between these two asthma phenotypes. However, we found that the significance was stronger in childhood onset asthma. The next part of the study was to try to have an even higher level of understanding of the mechanisms. For that, we separated the 113 genes significantly associated into 2 groups, those associated with these 2 asthma phenotypes. And we performed a tissue-specific enrichment analysis of the genes. We found that genes were in these 2 phenotypes were differentially expressed in blood and small intestine, suggesting a shared immune cause for the 2 groups. For childhood onset asthma, we found that genes were preferentially expressed in the skin, highlighting this tissue as a primary target for this group. And the adult onset results suggested a more lung-centered cause for this asthma phenotype. So the summary of the findings, this was the first large-scale genetic study to provide evidence for a greater genetic component in early onset asthma. We found shared and also distinct mechanisms. For example, results suggested that the establishment of the disease in childhood was driven by impaired barrier function in the skin and other epithelial surfaces, whereas in adult onset asthma, the cause were most lung-centered and environmentally determined. So this suggested that more effective treatments might differ between these two groups. Precision medicine strategies should account for age of asthma onset. So we perform a GWAS, a TWAS, and then this tissue-specific enrichment analysis where we separated genes into these two groups. However, you can imagine that we can obtain these groups of genes considering networks of functionally related genes. That would add a little bit of complexity in how we capture these mechanisms, these cellular mechanisms. And if we talk about networks, we have to talk about the Omnigenic Model for complex traits. This was a conceptual model proposed a few years ago, which tried to answer what we observe in GWAS, these small effect sizes that accounted for just a portion of the trait variance and was limiting our ability to understand the molecular mechanisms of disease. So what this model posits basically is that gene regulatory networks, as you can see in this figure, are so highly interconnected that potentially all genes expressed in disease-relevant cell types might be directly or indirectly affecting the disease. So in particular, the model proposes two kinds of types of genes. First, the core genes, which are these black stars that you can see here. These core genes are thought to directly affect the phenotype, whereas the peripheral genes are those that indirectly affect the phenotype by modulating the core genes. So the idea of the model is that these core genes might represent more attractive candidate drug targets. So how can we incorporate these ideas into our models? One idea would be to use gene co-expression networks. We could use the large RNA-seq data that we have, where we have measurements across different cell types, tissues, cell types under different stimuli. For example, here we have a group of genes expressed, for example, in macrophages under a viral infection, for example. And here we have another example of genes expressed in the heart. So the basic idea would be to capture these gene modules using some unsupervised learning method. A gene module is a group of genes that have a consistent or similar profile of expression across the same cell types. So you can imagine in this particular example that we have genes with different degrees of membership to each module. For example, in module 1, we have gene A and gene C, which are more strongly connected to the process than the other genes, for example. In this case, we downloaded these models from the multiplayer study. They applied player matrix factorization approach on recount 2. So the idea here is to connect TWAS results where we have associations for thousands of different traits and diseases with this gene module perspective, where we basically have two things. First, the relationship of genes. And second, we have information where these genes are expressed. And we also want to understand if these modules are associated with different diseases. In this example, you have two different, for example, processes that are affecting a cardiovascular disease. So we call this method phenoplayer. You can read about it in this preprint. The first thing that we propose is a regression model to compute a module trait association. So on the left, we have the gene weights for each module. And on the right, we have the gene trait associations from TIWAS. So the idea is to predict these gene associations for a particular trait using the gene weights of the gene modules. In this case, it's important to model the error terms using the correlation of predicted expression. And what the model basically tests is whether genes that more strongly belong to this module or have a higher weight tend to be more strongly associated to a particular trait. The result is a table like this one, where we have the p-value and the strength of association. This approach could be more robust to identify genetic risk factors. For example, if you see these two genes here, genes C and D, you can see that they are associated with this cardiovascular trait, disease, and they are also part of this mechanism here in this module. However, gene A, in this case, is not marginally associated to the trait, but it's still part of the module. So maybe this gene A is a key player. It's a core gene, for example, in the Omnigenic model that is not captured by standard methods. Later, I will show you an example of why we believe that that could happen. A second thing that we did in the paper was to learn shared and distinct transcriptomic processes across traits. What we did basically was to project the TIWAS associations into this latent space and then perform cluster analysis to understand these modules that were affecting similar clusters of traits. A third thing that we did to really try to test this idea of gene modules in genetic studies was to reuse an existing drug repurposing framework that we found to see whether integrating drug-induced transcriptional profiles into this module perspective was useful to predict drug disease links. So basically, we projected in addition to the traits into this space, we also projected these drug disease associations and predict drug disease links that we knew were FDA approved, for example. So let me show you the first part, the prioritization of candidate therapeutic targets. The first thing that we needed to have is a set of genes that was strongly linked with a particular trait of interest. In this case, it was lipid accumulation. For that, we collaborated with a lab. They perform a fluorescent-based CRISPR screen in the HepG2 cell line. Here you have two groups of cells. In the middle, we have cells with a viral transduction, where they diluted a fluorescent dye that was useful to stain lipid droplets. We identified 462 genes linked with lipid accumulation. In particular, we worked with this high-confidence lipid increasing gene set, which had 6 genes, as you can see. Then we performed gene set enrichment analysis and found 15 nominally enriched gene modules of our 987 modules in total. And the top module that was nominally enriched was this module that we identified with LV246. It was a module well aligned with pathways of lipid metabolism. We observed that the samples projected into this module were highly expressed in adipose tissue, which makes sense in this context. And we also observed the projected TIWAS associations into this module. So in this figure on the right, we have the top 15 genes of module 246. In the x-axis, we have the top traits after the projection into this space. And each square here shows 2 things. First, with a darker blue is the strength of the association between the gene and the trait. And with a small circle, we observe the colocalization probability. That's another result that gives us evidence of an association between a gene and a trait. It's basically the overlapping of the GWAS and EQTL signals. We also used a regression framework to test for associations, and many of these lipid-related traits were significantly associated. However, you can see that these two genes here, DGAT2 and ACACA, are part of the high-confidence gene set that we found with the CRISPR screen. You can observe that they are not associated to these lipid-related traits, and they are not even colocalized. So why is that? One explanation could come from the Omnigenic model. Again, maybe most of the genes in this module are peripheral genes, whereas these two genes are core genes that are directly affecting the phenotype of interest. Why do we believe that? We know that with TWAS, we only capture the cis-EQTL component of gene expression. That means that we only consider genes that SNP, that are close to the gene. So if we assume that the TWAS models for these two core genes that we believe are core genes are capturing all the common cis-EQTLs with no rare SNPs in the gene, then we can maybe assume that the gene module perspective is inferring these trans effects. And actually, the rest of the genes are modulating these core genes. And that's a reason why we are not capturing those two genes in this case that directly affect the phenotype. So this shows that the phenoplayer approach could improve the prioritization and be really interesting in this area to propose new candidate therapeutic targets, while also providing a mechanistic insight at the module level. So let me show you how we use this approach to learn shared and distinct transcriptional processes across traits. The idea is that gene modules representing transcriptional processes might be affecting different clusters of traits, and we wanted to understand that. For that, we use PhenomScan, a large TWAS resource that we built on the UK Biobank and other cohorts. We projected this data, as I explained before, into this latent space. Then we generated different points of view of the data to have more diversity. In particular, we use PCA and UMAP. We fed those representations into different clustering algorithms with completely different assumptions on the data structure. And then we generated an ensemble and derived a similarity measure between traits using the ensemble. So we generated, using a consensus clustering approach, different groups or partitionings of the traits. And the next step is the most challenging one in any cluster analysis application. You can see that the input data underwent a very high nonlinear transformation. But what we want to do is to understand which, in this case, modules are driving these clusters. So what we did here was to train different decision tree classifiers using the clusters as labels and the input data as training data. This allowed us to identify the gene modules that were separating these clusters of traits. And then we used those modules to test for replication in a TWAS resource built on top of eMERGE data. This was built in collaboration here at Penn. This is a sample of the input data, a heat map, where we have the 3 clusters in the columns. You can see that we have an asthma cluster, anthropometric traits, 3 clusters of autoimmune diseases. And we have the gene modules that were able to separate at least one of these clusters in the x-axis or as rows. We found that some modules were specific to some traits. For example, modules expressed in early differentiation stages of erythropoiesis were significantly associated with assays on red blood cells. Something similar happened with platelets and modules expressed in thrombocytes. In this case, in the autoimmune cluster, in one of them, we have these three hypothyroidism traits that were highly associated with a gene module expressed in the thyroid. So the approach seemed to be useful to facilitate the identification of shared mechanisms between diseases. We also tested this idea using a previously published drug repurposing framework. The idea of this framework is very intuitive. What we do is to compute a drug disease score using the gene trait associations and also drug disease associations from drug-induced transcriptional profiles. So an example of a positive association between a drug and a disease is like this one, where if you look at gene 1, we have a Z-score from TWAS of 5. That's a positive Z-score. That means that a higher expression of the gene 1 is associated with a disease. When I say expression, I mean predicted expression. But from the drug-induced transcriptional side, we have a negative Z-score. And that means that the drug lowers the expression of that gene. And that could be evidence that there is a link between the drug and the disease. So if you sum all these across different genes, you could potentially predict. Actually, the authors here show that that works. So that works for neurological diseases. So we did exactly the same, but using gene modules. An additional benefit here is that gene modules are interpretable genetic features. For example, we know in which cell types they are expressed. We used a gold standard with 322 drugs across 53 diseases to evaluate the prediction performance. We found that the module-based approach that we proposed was performed better than a single-based approach that was used previously, and you can see that even the prediction performance was slightly better. It was striking for us because what we are doing here is to basically project this input data across 22,000 genes into this reduced dimensional space. So reducing dimensionality might imply information loss unless the low dimensional space captures biological meaningful patterns, which seems to be the case. Then we took a particular drug and analyzed why this was working better. So niacin in this case was predicted as a disease-modifying indication for cardiovascular diseases. In this case, we took the score and decomposed this across all the gene modules. We found that this LV246, which I talked about, was aligned well to lipid metabolism pathways expressed in adipose tissue. This module was among the top 20 modules contributing to the niacin prediction of several cardiovascular diseases. However, here we know that this module is expressed in adipose tissue. That allowed us to hypothesize that maybe this is an indication that this module is representing a known mechanism of action of niacin, in this case in adipocytes. We also found another module that was expressed in immune cells with the same pattern. It was in the top 20 modules of several cardiovascular diseases. And that could be an indication of a mechanism of action that is also known of niacin on immune cells. So maybe PhenoPlayer could be also useful to provide an interpretable framework for drug repositioning. We are working now here with several faculty at Penn, especially with Dr. Blanca Himes, to understand the similarities and differences at the transcriptional level with between asthma and COPD, also with Dr. Peter Merkel on vasculitis and circadian rhythmicity with Dr. Karsten Skark. And I'm really interested in forming new collaborations to apply these different disease domains. So a little bit of my future research. My goal is to develop advanced computational methods applied to human diseases with a system biology approach. My research approach will be to use or develop, when needed, advanced machine learning methods for knowledge discovery. This will enable a multi-omics approach to precision medicine by integrating different data modalities. My immediate goals are to improve first the latent gene expression representations. I want to train new unsupervised models optimized for prioritizing potential drug targets. Also identify the optimal model parameters that are useful for specific diseases and not just for a global analysis as I presented today. I also want to change or challenge this gene module definition by learning alternative disease-relevant modules from TWAS, in this case, a different data modality than expression. I want to apply advanced clustering techniques to identify groups of genes with similar associations, and I want to integrate this in a later stage with gene expression data. And finally, I want to use this improved latent representations to build portable polygenic risk scores across different population ancestors. So the idea is to train these PRSs using these latent gene expression representations and then assess performance across different population ancestors. Given my previous research experience, I want to apply initially this to asthma, but the approach will be extendable to other complex diseases as well. So let me break down these aims. I will be presenting this with more detail tomorrow. So the concept of gene module is key in the phenop layer approach, as you saw in the presentation. I showed you today examples of very clear module signatures, like this one, where the expression is very clear, the metadata is great. However, that's not the case. That's not always the case. Sometimes the interpretation is hard for some modules, or as I said, the metadata is not really useful for interpretation. So in addition to that, there are other limitations of current models. For example, they could be not the best ones for the integration with genetic studies. There are also larger expression compendia today. It would be nice to test or obtain these representations from those outdated compendia. So I want to train new models optimized for the integration with genetic studies by varying the number of modules and other important parameters that could capture different genetic architectures of complex traits. Another limitation is that the methods that we were using are linear only. They only consider linear combinations of genes. So a natural approach here would be to use some deep learning models to capture these representations. However, we also want to test a method that we are developing here at the university named Cluster Match Correlation Coefficient, or CCC. We apply this method to gene expression data. We found gene pairs with nonlinear patterns identified by these coefficients in GTEx, as you can see in these examples. These patterns, or many of these patterns, were missed by linear-only coefficients like Pearson or Perelman. We also found that gene pairs replicated in tissue-specific gene networks from Giant, showing that these non-linear patterns in between these two genes are potentially part of the same process. And we also found these interesting patterns. For example, we have here genes UTY in chromosome Y and KDM6A in chromosome X, which replicated in all GTEx tissues and is explained by sex. You can see here that this very strong linear relationship are some male samples, and the orange ones are females. So this really highlighted the importance of considering sex as a biological variable, and cluster match was able to robustly identify these patterns. And of course, this is not limited to gene expression data. This is a general correlation coefficient. It could be useful to find these patterns in other data modalities, like electronic health records, for example. It would be great to apply that in Colorado. Then what I want to do is to learn alternative disease-relevant gene modules from GWAS. During this presentation, I always use this definition of module, which is genes in modules have similar expression across profiles across the same cell types. However, a potentially different definition is that genes in modules have similar marginal associations across the same traits. So what I did here is to apply a biclustering method. I will give more details tomorrow on PhenomeScan, our GWAS resource. And I found that these gene biclusters, with in this case, with an asthma diagnosis, were significantly enriched for different gene ontology terms, suggesting that this is probably a valid alternative way of finding these gene modules. Then I want to use these improved latent representations to develop portable polygenic risk scores across different population ancestors. Very briefly, a PRS predicts an individual's genetic liability to a disease. So when we apply these methods to a cohort, we generate scores for all the individuals having a distribution like this one. So if one individual is in the increased risk, that could be helpful to make better decisions about a person's health. For example, if the high risk is for a cardiovascular disease, maybe it could be suggested to change the diet or some lifestyle parameter. So PRSs offer great potential as clinical instruments, and it's of a special interest here in Colorado, where I participate in this clinical polygenic risk scores for use in translation. It's a working group led by Dr. Christopher Guinou, and it will be fundamental for me to present here and get feedback and test these ideas. However, there are some issues with PRS, and that's why I want to do something regarding that. This is the way we compute polygenic risk score. Basically, we take the genotype of the individuals using a set of SNPs. We use an estimated effect size for that SNP. We obtain this this effect size from GWASs, public GWASs, for example. However, the problem is that most GWAS are conducted in European populations. So these effect sizes are estimated only in European populations. And that's the reason why PRSs have poor performance in non-European ancestry populations. So if you imagine a widespread implementation of these tools, that could really exacerbate race-based health disparities. So how can we improve this? This is the variant-based PRS I was talking about. We recently proposed polygenic transcriptome risk scores, where the basic idea is to use predicted gene expression instead of variants. And we show that this improved the portability of polygenic risk scores across ancestries. We use, in this case, 17 quantitative traits, and it was the performance gain was most clear in African Americans. Another idea is to use gene modules and build a gene module-based PRS, where instead of variants or genes, we use these groups of genes that are functionally related. So the basic idea would be to first predict gene expression using TWAS, which is something that we can perfectly do now, and then project this data into this gene module space and train the PRS here in this space. I believe that this could work because my hypothesis is that gene modules might be more conserved across individuals, improving actually not only portability, but potentially also the stability of these scores. And that could improve improve the portability across population ancestors. I'd love to test these ideas using the CCPM biobank. As I said before, my participation in the PRS UIT will be fundamental. Actually, this is part of my the root phase of my K99. So I can't wait to start testing these ideas. I have to say thank you to so many people. First of all, Casey Green and his fantastic lab. Also my mentor here at Penn, Marilyn Ritchie, Dr. Hakim from Chicago, and Georgina Diego, my PhD advisors. Thank you so much for listening.
Thanks so much for that wonderful talk, Milton. There's time for questions.
I guess I'll step up and ask a question. So, hey, Milton, how's it going?
Hey, James.
So, you talked a little bit about the use of gene modules in various methods, and you talked about, a little bit about modules in general. But I'm kind of curious because that I know you tried to define some of this, but the idea of a gene module is very squishy. And genes can go in, genes can go out. So how are you thinking about the definition of the module in this case, particularly when you were talking about the last set where you're trying to project the features onto the actual modules themselves? I'm just kind of curious in how you're thinking about if you're thinking about developing better gene modules, maybe like tissue specific module, like how I'm just kind of curious on expanding upon that idea.
Yeah, that's a great question. As I said, the concept of gene modules is fundamental here and also in this proposal. So that's why, first of all, what I want to do is to use existing methods to try different alternatives in learning these modules. For example, well, today I was talking with Sean Davis. He has a method called genomic supersignature, which uses a completely different, I think it's a completely different approach. The prior acknowledged information is plugged differently in the model. The PCA component is applied on each data set individually. So that's one way of trying this, trying different methods with different parameters to learn these models, taking care of also the technical effects, for example. And also, I also want to try a little bit models that are a little bit more probably robust. We are finding that this cluster match correlation coefficient could be really helpful here, finding more robust patterns or deep learning models also. So yeah, I think we will generate different alternatives to really test this idea based on the Omnigenic model. I expect that this could work for some diseases. Actually, what I expect is to see what we saw in the drug prediction task, where in general, the idea worked. But if you look at specific disease domains, you can see differences in performance. So maybe even a combination of gene modules with single genes could be a good alternative here.
Yeah, I mean, I think I guess you can go in many directions with that. And I'm curious on, you talked about finding robust modules, and I wonder, this is a hypothesis, I guess, if the robust modules are the best ones to project on. I mean, if you have disease specific or treatment specific or different types of modules that are responding differently under different conditions, those might be the more interesting ones. But it's an open area for exploring. So.
Mm-hmm. Yeah, I agree that this is a key point. I mean, this will be very important.
I have a question. It looks like somebody in the chat also had a similar question. But why is it that the like the transcriptomics based risk score would be more portable across ancestries?
Well, one idea is, as I said, that maybe the predicted expression is when I say predicted expression, I mean the genetically predicted expression, the expression that we can predict using genotypes. When you take one model, for example, one model for a gene, you have several predictors, several SNPs that are predictors of this expression of the gene. One way to explain a potentially more conserved feature of this predicted expression is that you use several SNPs, maybe across different individuals or ancestries. Those SNPs are slightly different, but still enough to predict reasonably well the expression of the gene. So that would make the model more robust than using individual SNPs. And maybe the idea of a gene module is a step higher in this idea of robustness, and that could potentially work. Did I ask your question?
Yeah, I think so. But also in your, so you had that slide where I had like, a PRS and, it's kind of the sum of the SNPs. And then the next one was the TPRS. Is that predicted transcriptomics or was that just based on TWAS?
That was, I want to share again the,
okay.
Yeah, let me go there. So the PTRS, can you see the PTRS, this one, right?
Yeah, the PTRS, yep.
What we did here was to use, this is the predicted expression of one gene in one individual, and this is the effect size computed using an elastic neck model. So what we are using actually is the predicted expression.
Okay, got it.
Okay, perfect. Great.
Thanks.
Sure. Thank you for the question. Is there any other question?
Oh, can I ask a follow-up question?
Sure.
Yeah. So here I know your PTRS, the transcriptome is predict from the SNP, right?
Mm-hmm.
How do you think it's compared to the actual measurement of the gene expression?
Can you say it again, the question? Sorry.
Well, you say, I mean, here you propose to use the genotype predict the transcriptome, and then apply to a different kind of race to predict the disease risk, right?
Mm-hmm.
How does this kind of predict from SNP compared to the actual measurement of true gene expression? Use the transcriptome, to compare to do a disease risk prediction. Do you think
so?
Is there any risk difference regarding the gene expression?
There's a little bit of background noise. Sorry.
So why I say is, do you think there's any kind of difference for the race regarding the gene expression? We know SNP are different from the race, right? How about the gene expression?
I think that you might be asked, if I understood correctly, is that you say that the predicted expression could be different across different ancestries. Is that what you were asking?
Right, but because when you use TWAS here, you don't consider about race difference, right? You just put a SNP in and then using the GTEx or something, the database, and then to predict the gene expression. So which
Yeah, what we did was to, in this particular study, was to train models in Europeans populations, then predict risk in Europeans, get the prediction performance, and then use exactly the same models to predict risk in different ancestors. Basically, what we are comparing is the ratio of the differences of the performances in different ancestors. If I understood correctly, let me know if I didn't.
So you're when you're talking near the end about sort of identifying these nonlinear correlation structures, and you were thinking about kind of applying them in an EHR context. Could you give kind of an example of something that may be interesting in that?
Yeah,
in that kind of context.
Yeah. Let me go back a little bit. Yes. So this is, I think this is a nice example in gene expression data. Here we have two numerical variables. However, the cluster mark correlation coefficient can work also with categorical data, with multiple categories. So you can imagine, for example, finding in EHR these clusters of samples, for instance, in this case, with sex differences that we are seeing that are completely missed by standard coefficients. So, for instance, you could have, I don't know, maybe blood pressure, different diseases, and completely non-linear pattern that is meaningful if you separate samples by sex, for example, but it's completely missed by a linear correlation coefficient. So I imagine that could be a potential application. It could be, of course, necessary to talk more with someone that has more expertise in this area, but I believe that could be a potential application.
Anyone got any more questions for Milton? Well, if not, thanks everyone for joining. Thanks again, Milton, for giving a wonderful talk.
Thank you so much, everyone.
Bye.
Bye-bye.